Hello everyone, my name is Aarat Kalra. I am a PhD student at the University of Alberta, working under the supervision of Professor Jack Dyczynski. My work across my PhD is on the electrical properties of tubulin and microtubules. Now, before I begin describing to you my work, I just thought I'd introduce microtubules and tubulin a little bit, and then we can go into their electrical properties. So microtubules are these slender polymers that are present ubiquitously across the cell. If you were to zoom in on one of these polymers, you'd get a structure that looks like this. Okay, and so you get a cylindrical cross section. If you were to zoom in on a microtubule, you'd get a structure like this, which is tubulin. So tubulin makes up microtubules that are then present all across the cell. So microtubules are these cylindrical filamentous polymers. Okay. Now, and these microtubules play a variety of intracellular roles. Okay, they're very important for the maintenance of cell shape and rigidity. So they push against the cell membrane. They're crucial for cell division, forming the metaphase plate, so mitosis. And they also act as these as basically as intracellular as intracellular railroads for the transport of macromolecules. So what happens is, if you want to transport a mitochondria from point A in the cell to point B, you do it along a microtubule. So you'd have these motor proteins, the kinesin and dynein, that literally carry a mitochondria using microtubules as a substrate from one place to another place, and that's what makes microtubules act as railroads within the cell. So they act as guides for the transport of massive membrane-bound organelles. Okay, microtubules play a variety of other intercellular roles as well. All these roles that are presently well researched and now beginning to get understood are primarily biochemical and or biomechanical. Now, what has been so far less well researched are their electrical properties. Okay, now what do I mean by that? So the thing about tubulin is that it has very interesting properties, not only from a biological perspective, but also from a slightly physicsy or an electrical engineering perspective. Okay, so it has an extremely high negative charge. Okay, so this is a histogram of 12,000 proteins and their charges. Here we see that the most proteins peaked at about -5. I think this will come out to be something between 0 and -5. Okay, that was the charge that was highest. Tubulin, on the other hand, had a charge of -50, so it's extremely highly negatively charged. Not only that, it has a very high dipole moment. Again, it's pretty much off the peak over here. The peak is 400 dBi. Okay, tubulin's dipole moment is about 1800 dBi. So it has a very high charge and a very high dipole moment. And these are important things to remember because they'll play up throughout this talk. And this is what makes tubulin very interesting from an electrical perspective. Okay. Now, because of the extremely high negative charge, what you have is, if you have microtubules just roaming around randomly on a bed of kinesins in solution, governed basically by thermal movement, and you apply an electric field like so, the microtubules all reorient and they move towards the positive electrode because they're negatively charged, right? In the presence of an AC electric field, you'd get microtubules basically aligning to the external electric field lines of force. So here you can see microtubules that are initially perpendicular to the electric field lines of force now reorient. So what happens is, you have a plus electrode here and a minus electrode here. A microtubule that was initially oriented normal to the electric field lines of force would now flip. Much like this is very similar to what would happen in an electromagnetic in a ferromagnetic material when you dipole flipping of a ferromagnetic material. So this is what happens in the presence of AC electric fields, and so because of their electrical properties, microtubules have also been modeled to act as wires within cells. Okay. Now, why would they act as wires within cells? That sounds like a very counterintuitive thing, right? Well, they would act as wires within cells because of their negative charge. You see, because of the negative charge, microtubules attract a family of counter ions to their outer surface. Okay, so you have these polymers that now attract these, they basically have a coating of cations on their outer surface. Okay, these ions are modeled, of course, they are very high density, in front of, right next to a microtubule simply because they're extremely negative charge. But in the presence of an external electric field, these ions would move around a microtubule. And so the entire microtubule would act as a wire because it's transporting charge or current when exposed to an electric field. And so and there's a lot of papers on this that mathematically model microtubules acting as wires within cells, basically for ionic transport on their outer surface. Okay. And so the obvious question is, what are the electrical resistance and capacitance of these wires? Is the resistance low enough for them to even be called wires, or is this just a fancy theory? What is the capacitance of these wires? Capacitance is the ability of something to hold a charge. Okay, so it's basically defined as charge held per unit volt. Okay, now normally you would characterize the electrical properties of nanowires such as microtubules using solid state impedance spectroscopy. So this is what was done for carbon nanotubes. Thing is, you can't really have microtubules in solid state. Okay, because as soon as you take microtubules out of water, out of the buffer, what happens is microtubules compress. So the cylindrical cross section that I talked about becomes flattened, you see. And so microtubules are not really in their native condition when they're outside water. Okay, and they'll additionally denature and things like that. So you can't really take microtubules outside the buffer, which is what you do for carbon nanotubes. The other thing you can do is you can measure, so you can put microtubules in the buffer and measure the electrical properties of these ions in a very low ionic strength, low microtubule concentration environment, and then try and glean the information of how the microtubules change the electrical properties of the entire solution. But you see, the ionic strength in our bodies is not really low, and neither is the microtubule concentration. In fact, in our bodies, in cells, the tubulin concentration varies between 20 to 120 micromolar, so it's extremely high. It's not in the nanomolar regime. And also the ionic strength is very large. It's at least more than 100 millimolar, at most 600 millimolar. Okay, so compare 100 millimolar and 600 millimolar to 20 millimolar, which is what you typically use in a low ionic strength buffer. So my first experiment, when I sat down with my supervisor, was to mimic intracellular conditions, which is to say this stuff, and see if microtubules changed the electrical properties of this solution, of a high ionic strength solution. Does the resistance of the solution increase? That would mean microtubules are acting as anti-wires, right? As the opposite of a wire. Does the electrical resistance of a solution reduce? Charges are transported more easily. If they are transported more easily, maybe they do actually act as wires, for ionic transport. And so that was what we were starting off with, just a very simple, idea. And I went about doing this using some what are called FTO coated glass slides. So I cleaved them into broad and thin morphologies, as you can see over here, and I put some double-sided tape on top. And then I put the thinner FTO coated glass slide on top of the broader one. Okay, so you have this side view. Okay, so you have the broad FTO coated glass slide here and the thin one on top. And the thing about FTO is fluorine doped oxide is conductive. Okay, so this layer over here is conductive, as is this layer. Okay, and so you have what physicists call a parallel plate capacitor geometry. Okay, and so what happens is you can put something in between, say your buffer, and you have a parallel plate capacitor geometry that determines the electrical properties of this buffer. This is a very well studied system. Okay, and then you can glean things like properties such as resistance and capacitance of whatever material is between the parallel plate capacitor. Okay, so you can connect this to electrodes and things like that, and actually measure the electrical properties of the solution. Let me remove these. Okay, so here you see me actually flowing in some solution. I don't know if you can make this out. You're flowing in some solution into this contraption, into parallel plate capacitor, and you can wick it out using a basically these strips of filter paper. Okay, and so you basically now have created a solution exchange setup, right? You can this is a technique that I borrowed from my master's that used turf microscopy, total internal reflection fluorescence. That's what they do over there also. So this is basically using that technique, but for a different application. And once you have buffer in the within the plates of the parallel plate capacitor, you can then you can then actually just connect this to a 3 electrode setup and read off the values, and the values will give you the resistance and the capacitance. Okay. Now, before you actually proceed to measure the resistance and capacitance of microtubules, what you have to do when you build such a system is you have to do controls. Okay. So we measured the imaginary component of impedance, which is the capacitance in these low frequency regimes. Okay. So I'll just call this capacitance, and you measure the real component of impedance, which is also known as the resistance. Okay. And you do this for a variety of electrolytes, so things like KCl, CaCl2. They are very well studied, and only when our results concurred with previous literature did we actually go forward, and we knew that our setup was actually working. So we felt confident of actually now measuring the electrical properties of microtubules, now that we had established the capacitance and resistance of these solutions, and they were mirroring that of those found in previous literature. So we went ahead, we polymerized microtubules from Tubulin. We polymerized microtubules at 3 different concentrations. So it was 0.22 micromolar, at 2.2 micromolar, and 22 micromolar. Note that most experiments have been done on 0.22 micromolar on the electrical properties, but the concentration inside our bodies is at least 20 micromolar, right? It's between 20 and 120 micromolar. So there's a big difference between what has been experimentally done, which is 3 orders of magnitude off, basically, at least 2 orders of magnitude off, compared to what actually exists in our bodies. So I wanted to have these 3 different concentrations, 0.22, 2.2, and 22, to really just understand how microtubule concentration affects the electrical properties, as a network. Okay, so you do the experiment. You flow microtubules into the parallel plate capacitor, and you have the background buffer, which is called BRB80, which is Bradley's reconstitution buffer in 80 millimolar pipes. So that's the background buffer, and you have your 3 microtubule concentrations that you solution exchanged, and you read off the values. I also did the same thing with tubulin. Okay, so I had exactly the same concentration of unpolymerized tubulin. So 0.22 tubulin versus 0.22 microtubules. Okay, 22 microtubules versus 22 tubulin. And now you can compare not only what happens to microtubules as a function of concentration, we can also compare what happens to the electric properties as a function of tubulin versus microtubules. Okay, so you can compare unpolymerized to polymerized tubulin in this method. Okay. And yes, you can subtract the background from the microtubule solution, and you get these readings. So for 0.22 micromolar, we found that microtubules increased solution capacitance, whereas unpolymerized tubulin, which is the red line, didn't really do much. And you can see that this behavior for unpolymerized tubulin is pretty much the same. You can crank up the concentration by 2 orders of magnitude, but really it makes no difference. The tubulin does not alter the capacitance of solution significantly, whereas microtubules, on the other hand, increased the capacitance of the solution. The most prominent this effect is seen most prominently at 22 micromolar, which is close to the intracellular tubulin concentration. So here we see that microtubules actually increase solution capacitance, whereas unpolymerized tubulin at exactly the same concentration does not. Okay, so that's what we saw in the imaginary component of impedance. So after analyzing the imaginary component of impedance, you can also perform exactly the same analysis on the real component of impedance, right? So this is the resistance basically. So you compare the properties of microtubules to those of tubulin at exactly the same concentrations. And so if you take a look at that, we find the answers to that are actually less clear. You can see that in almost all cases, microtubules and tubulin, red line, blue line, they seem to overlap with each other. The error bars are very high, and the results are largely they seem to be the same for both microtubules and tubulin. All you can say is we found that for the intracellular case, for 22 micromolar, both microtubules and tubulin increased solution resistance. Okay. However, interestingly, if you were to zoom in just between 10 and 100 hertz, okay. So if you have this graph and all you do is zoom in between what you got for 10 and 100 hertz, you'd find something very interesting. You'd find that at this point in this range, the resistance contribution of microtubules was actually very interesting. You see, there is within this range, the delta Z is actually negative. So the value of resistance, the value of microtubules minus buffer is actually negative, which means microtubules were actually reducing the resistance in this regime. And you see that across all concentrations. Do you see what I mean? Between 10 and 100 hertz, it's always negative, the delta Z. This does not mean that the resistance is negative. This means that the difference in resistance is negative. So microtubules minus buffer is negative, which means microtubules are actually conducting in just this regime, 10 to 100 hertz, and then they resist. They're more resistive than background buffer once again. Now, of course, this, as I mentioned, this is an extremely high noise experiment. So before we conclude that there is actually a flip in resistance between 10 and 100 hertz, more experiments are required, maybe using a different technique, perhaps using even a third technique that's orthogonal to this. But we found that for the same concentrations of tubulin, so red lines, this behavior was actually not mimicked, which indicates that maybe there is something going on between 10 and 100 hertz. But like I said, more experiments are required to confirm a flip between 10 and 100 hertz. Okay, and so once you have the resistance and capacitance of your microtubule and tubulin network, what you can do is you can fit it to an effective circuit, to an equivalent circuit, and see what microtubules and tubulin are best modeled as. Okay, so we fit it to a whole bunch of circuits, and we found that an RC circuit in parallel was actually the best model for a microtubule network. And you can see that the fit is quite good, except of course for this 10 to 100 hertz regime, which like I said, was an interesting point where the microtubules actually reduced the resistance of the solution. So that's what we saw. When you fit it, you get fit parameters that indicated that the microtubule capacitance, the network of the microtubule network's capacitance was in the microfarad regime, and the microtubule network resistance was in the kilo ohm regime. Okay, at intracellular concentrations. So this is an important result because we were the first people to measure the capacitance of microtubule networks, and we were the first people to do so within intracellularly relevant conditions. And because of these reasons, we even actually were lucky enough to get our data published earlier this year. So you can look that up if you like. It was published in the journal called Nanomaterials earlier this year. And our work seems to indicate that microtubules may act as charge storage devices, so intracellular capacitors within the cell, because of their large capacitance in the microfarad regime. And so the electrical properties of microtubules, while not significant as a resisting wire, are actually significant as an intracellular capacitor or a holder of charge, so to speak. And yeah, so with this, I thank you for your attention. I'd like to thank members of my group for all their support, and thank you for listening.
I'm Chris Rourk, and I will discuss conductive atomic force microscopy, or CAFM, testing of human substantia nigra pars compacta tissue, or SNC tissue, and indications of electron transport from those tests. First, I will discuss the hypothesis that ferritin and neuromelanin form quasi-ordered arrays that function like quasi-ordered arrays of quantum dots. Next, I will review CAFM test results on SNC tissue that are consistent with the hypothesis. They do not prove the hypothesis, but if the behavior of the tissue is consistent with the hypothesis, then that is a significant fact. I'll also discuss very briefly the relevance of this to consciousness. What is the evidence that ferritin and neuromelanin in SNC tissue could support energy transfer? Ferritin is a common iron storage protein formed from a combination of light and heavy chain protein units. Inside of the protein shell is a ferrohydrite core. The structure is similar to that of a quantum dot, which can be modeled as electrons that are stored within a Coulomb blockade. The electrons are prevented from being conducted out from the quantum dot by the Coulomb blockade and instead have to tunnel or hop. This is an animation of a molecular dynamics model of an Fe2+ ion migrating out from the ferritin core through the shell, protein shell. The release process is associated with acidic pH conditions, and the storage process, where the Fe3+ ion is stored as Fe2+ ions in the core, is associated with basic pH conditions. The electrical properties of an individual ferritin core results in a nonlinear current voltage response that is flat at low voltages, and that is similar to the current voltage response of individual semiconductor quantum dots. As you can see, the current at low voltages is essentially zero. In this configuration, the ferritin was compact instead of being separated, and was somewhat structured in these linear arrays, which resulted in high conductivity regions at certain voltage levels. As I will show, this conductivity behavior is consistent with the way quantum dot arrays behave. This device was reported in a paper by Barra et al., which I will refer to as the Barra device. In this configuration, layers of quantum ferritin were deposited. And the current was measured from the top to the bottom layers, as well as across the layers from side to side. As can be seen, there was relative disorder within each layer, but the layers were ordered in 2 dimensions along the X and Y axis. From top to bottom, nanoamperes of current were measured in a roughly linear relationship up to 4 layers in thickness. Above 4 layers, no current flow was measured. However, from side to side, microamperes of current were measured in a logarithmic relationship. That is roughly 3 orders of magnitude greater than the top to bottom measurements, even though there are roughly 3,300 ferritin cores in series between the 2 electrodes, compared to 4 ferritin cores in series from top to bottom. This behavior is consistent with the electrical characteristics of quantum dot arrays and cannot be explained using non-quantum mechanisms, which I will refer to as classical. Keep in mind that individual ferritin cores do not conduct at voltages of less than 0.5 volts, as we've seen. The voltage difference between cores from the side to side with an applied voltage of 3 volts at the electrodes is less than 1 millivolt, which should not result in any appreciable current if the current behavior was classical. Ferritin does not leak ferrohydrite, and it requires an acidic pH to transfer Fe2 ions from the core and to convert them into Fe3 ions outside the protein shell. So this behavior could not be due to ferrohydrite outside of the ferritin core. The drawing on the left is a 4x4 array of quasi-ordered quantum dots. The computer-modeled effective conductivity curves are shown in the middle, although this is not classical conductivity, but rather electron transport. The solid line shows the results if the quantum dots were to have a uniform size and spacing, and the dashed line shows the effect of scattering due to the disorder of the quasi-ordered array, which broadens the conductivity curves relative to the ordered case and lowers the total conductivity, but it is still greater than 0. These conductivity curves of tunneling behavior are sometimes called electron minibands, and they show that the electrical behavior of ferritin in arrays is like arrays of quantum dots due to the electron wave function. The graph on the right shows calculated current voltage responses for much larger quasi-ordered arrays of different sizes, from 125 by 25 up to 2744 by 196. And these are similarly logarithmic above a threshold voltage. These studies demonstrate that quantum dot arrays behave differently than individual quantum dots due to the electron wave function. Now, we move from studies of ferritin in integrated circuits to studies of ferritin and neuromelanin in neurons. Photo on the left is of the cell body or soma of a dopamine neuron. There's a high concentration of neuromelanin in neuromelanin organelles, or NMOs, that are distributed throughout the soma. NMOs are autophagic vacuoles, which are organelles that essentially break down waste materials in the cell. They are present in many neurons, but are particularly large and plentiful in catecholaminanergic neurons like dopamine neurons. The center photo is a close-up of some NMOs, which contain roughly 30 nanometer spherical neuromelanin structures that are surrounded by other parts of the organelle. And the photo on the right was taken with electron spectroscopic imaging to show the ferritin, which is in red, and that is shown as in layers around the NMOs. These ferritin layers are structurally similar to the ferritin layers in the Barra device. The large soma of the SNC dopamine neurons have the most neuromelanin. In this photo of SNC tissue, staining shows where there's iron, which is mostly ferritin. The glial cells between those neurons, which are shown as the black dots, also contain a lot of ferritin in the SNC. The hypothesis is that all of these large neuron soma are coupled by the quasi-ordered ferritin and neuromelanin in each soma and in between the soma and glial cells, and participate in the electron transport mechanism. Glial cells include microglia, astrocytes, and oligodendrites, and they interconnect the neurons at various locations as a function of the type of glial cell. Glial cells are not neurons, but support the function of neurons. At least in the SNC, they contain high levels of ferritin and can provide additional ferritin structures between the soma of the neurons. Now I'm moving back from neurons to integrated circuits. These images show the design of a photovoltaic cell that uses quantum dots and pi conjugated polymers. These structures were designed to form excitons and to conduct the electrons in one direction and the holes in the other direction to create current. An exciton is an electron hole pair that can be formed when an electron gets enough energy to break free of an atom, and in this regard, quantum dots are considered to be artificial atoms. For an exciton, Coulombic attraction keeps the electron and hole together. These structures are similar in some regards to the distribution of ferritin and neuromelanin in SNC neurons, but they do not form quasi-ordered arrays, and they do not have sufficient order to support formation of the electron wave function minibands. Instead of the structure seen in the solar photovoltaic cells, which conduct electrons in holes between electrodes, the NMOs and ferritin in neurons can be modeled as layers of p-type ferritin that surround the n-type neuromelanin. Triplet state electrons, which I will address further, facilitate exciton formation and are represented here as free electrons. These structures do not conduct the electrons and holes of excitons to terminals, and instead allow the electrons to persist. Excitons have a Bohr radius, which is directly related to the relative permittivity, and the relative permittivity is much greater than 1 in neural tissue. The Bohr radius of an electron at room temperature is roughly 6 nanometers, and the increased Bohr radius extends the electron wave function and facilitates miniband formation. In one process, neuromelanin is formed from dopamine, which interacts with ferric ions to form dopamine quinones. These eventually form eumelanin and pheomelanin, which are both pi conjugated polymers. A pi conjugated polymer has delocalized electrons that are shared across adjacent aligned p orbitals, which can create conductor or semiconductor properties. The melanins combine with ferric ions to form neuromelanin. NMOs are also present in many other types of neurons and can be formed in a process from incompletely degraded proteins and lipids from the breakdown of mitochondria. They don't require dopamine to form. In addition to contributing to the formation of NMOs, dopamine also contributes to the formation of triplet electrons. The quinones that are formed from dopamine can also degrade the melanins into dioxetanes, which cleave at temperature, room temperature, to form carbonyl with a triplet state electron. Triplet state electrons are high energy electrons that are prevented from dropping to the next lower energy state because they have the same spin as the electron in that lower energy state. In melanocytes, a similar process creates triplet state electrons from UV energy that is absorbed by the cell. And those triplet state electrons can cause damage to the DNA contained within the cell nucleus, many microns away, in a process that occurs hours after exposure to the sun. This indicates that the triplet state electrons persist for a long time inside of the cell and that they are not rapidly neutralized. Now I will address CAFM test results, which I decided to commission and self-funded based on the evidence that ferritin and neuromelanin could provide the hypothesized energy transport mechanism. The ability of CAFM tests to detect this function of neuromelanin and ferritin in these neurons was predicted in my first paper. The tests were performed on fixed SNC tissue samples from AMS Bio, which is in the business of selling tissue samples for scientific research. They were shipped directly to EAG Labs in Sunnyvale, and Dr. Sarah Ostrowski performed the tests using a Bruker dimension icon atomic force microscope. I also asked Dr. Yuming Hua at Bruker to review the results, and he confirmed that they appeared to be accurate and not the result of equipment misoperation or contamination. These are optical images of the tissue that was tested. The image on the left is on glass, and the one on the right is on foil. CAFM testing requires a ground plane, but the glass slides were used to index the locations of the large SNC neurons. These were hard to identify due to a lack of staining. In order to keep the number of variables to a minimum, the tissue was not stained. The slide on the left is similar to areas that were tested for tests 1, 3, and 4, and the slide on the right is similar to the area that was tested for test 2. Because of the lack of staining and the lack of backlighting, it's not easy to make out these round structures, which are the large dopamine neuron soma on the left, but they're clearer on the glass-backed slides. CAFM testing uses an oscillating probe that's moved across the surface of the specimen in a raster. The height of the oscillating probe is determined using a reflected laser signal, and current and force can also be measured. The Bruker device generates 3 current data points per oscillation cycle, one for the entire cycle, one for the period of time during which the probe is contacting the sample, and one for the period of peak force. There are 512 cycles per line and 512 lines per test, for 262,000 individual cycles and 786,000 current measurements. The CAFM data can be mapped to help visualize what has been measured because of the large number of data points. This is a height map on the right, and it shows these thin areas that correspond to the dark spots have some material at the bottom of the thin areas. There's also a side view of a current map on the left. And what is notable about this map is the positive currents. Because the sample was grounded and a minus 10 volt bias was applied to the probe, the only currents that should have been measured would have been negative or flowing out of the node, out of the probe into the ground plane. This is using the convention that the direction of electron flow out of a node is negative current. Because the probe has a negative voltage, it is an electron source and should have only created negative currents into the ground plane. These positive currents represent electrons flowing into the probe against the minus 10 volt bias voltage, which would require a more negative potential to create. This is unusual and non-classical behavior. But the behavior makes sense in light of the structure of ferritin around NMOs, as we have seen. The oscillating probe with a minus 10 volt bias creates a field of 2 million volts per meter on the 5 micron thick tissue sample sitting on a foil ground plane. And that field strength is high enough to cause excitons to form in the tissue. These excitons can result in electrons that tunnel through these ferritin sheets that surround the NMOs to the probe. The probe provides a lower impedance path to ground than the insulating layer of the surrounding neural tissue to the ground plane. An electrochemical reaction would not cause this behavior because the probe tip is made of a highly stable material that is not chemically active, unlike battery terminals. Even if it was made of a battery terminal material, the largest battery reaction potentials are much less than 10 volts. The second test was in a location without any structural similarity to the large SNC dopamine neurons, as can be seen in this height map, which shows no round, thin areas, as was seen in the first test. A better wax removal protocol was also used that included rehydration of the tissue. However, even with this wax removal protocol, there was no positive currents measured in this location. This test location provided a control sample, though, and demonstrated that these positive currents were not artifacts of the test specimen or misoperation of the equipment. The negative currents were measured in thin areas. The third test yielded very significant results. There were widespread positive currents measured, with the zero current line being approximately here. There are many thousands of measurements above zero, which provide very compelling evidence that these currents are an actual physical characteristic of the large dopamine neuron cells and not a measurement anomaly. It should be noted that experiments have shown that solvents that were added to solid-state quantum dot arrays improve the conductivity and electron transport in those arrays. And this may also explain why there was an improvement in measured currents in this tissue after it was rehydrated. The fourth test focused on a smaller area of this sample, which is the third test, where there was a higher current measure, which is these lighter areas. The first three tests were on 78 square micron samples. So it was decided to reduce the fourth test area to a 2 micron square area. The approximate size and location of that reduced area is shown as a box here. And as you can see, it is in one of these light areas. These histograms also help to visualize the current data from the third test. You can see that the distribution of the positive currents, and there are a large number of them, changes over the peak current measurements and the contact average current measurement period, which is non-classical behavior. You would expect this shape to be the same with classical behavior. The fourth test also yielded very significant results. In particular, the mapping of the anomalous positive currents, which can be seen here, corresponds to structures having dimensions and arrangements of the ferritin, as in the sheets here, and the neuromelanin organelles, which would be these round structures here. This is consistent with the hypothesis that these biostructures are functioning as quantum dots. And this is a height measurement showing that these measurements do not correspond to surface features but are underneath the surface material. The current histograms for the fourth test also show positive currents and dynamic current behavior that is non-classical. The zero current points are aligned for these three histograms, but the scale on the lower one is different. How is this applicable to consciousness? This electron transport mechanism allows a large number of neurons to share a common state, because the electron wave function is a three-dimensional probability wave of where an electron could localize. It exists at all locations until the electron localizes. It is an integration mechanism that can integrate thousands of individual neurons using electron wave functions that overlap for a large number of electrons. The state of each neuron is determined by thousands of dendritic inputs to the neuron and almost a million of axonic outputs. Even if each neuron only represents two states, such as a participating and non-participating state, the number of different states would be very, very large, approximately 10 to the 300, which compares to approximately 4 times 10 to the 80 atomic particles in the known universe.
Hello, my name is Majid Beshkar. I am Assistant Professor of Oral and Maxillofacial Surgery at Tehran University of Medical Sciences in Iran. Also, my education and job are related to the field of maxillofacial surgery. Consciousness has been one of my primary research interests in the past 15 years. I have recently introduced a theory in an attempt to explain the hard problem of consciousness. The theory is named Qbit theory, and the first version of this theory has been recently published by Integrative Psychological and Behavioral Science. On the left side of this slide, you see the first page of my article entitled The Qubit Theory of Consciousness. I named this theory Qubit because it is a multidisciplinary theory that is primarily based on quantum mechanics, biology, information theory, and thermodynamics. I think that we can solve the problem of consciousness if we find answers to 4 questions. In fact, the Qubit theory formulates the problem of consciousness in 4 questions and attempts to provide preliminary answers for each question. In this slide, you see the first question. What is the nature of qualia? Here, the term qualia refers to subjective experiences, conscious percepts, or simply percepts. Qualia is plural for quali. According to the Qubit theory, a quali is a superdense pack of meaningful quantum information encoded in maximally entangled pure states. Therefore, qualia are information in nature. More specifically, they are quantum information, not classical information, and they are meaningful or semantic information. This is the second question of the 4 questions that comprise the problem of consciousness. How are qualia generated? The answer is that when a pack of meaningful quantum information is compressed beyond a certain threshold, a quali is generated. This answer emphasizes the importance of information compression in the emergence of consciousness. To generate consciousness, a very high degree of information compression is necessary, and classical physics does not allow for such a high degree of compression. For this purpose, we need quantum phenomena like entanglement and coherence. This is the third question. Why are qualia subjective? The answer is that a quali is subjective because a pack of information encoded in maximally entangled pure states are essentially private and unshareable. I think that the subjective aspect of consciousness is its most difficult aspect to explain. However, the quantum mechanical phenomena, such as entanglement, could help us explain this difficult aspect of consciousness. As described by Horodecki and his colleagues, entanglement is the quantum equivalent of what is meant by privacy. According to the monogamy principle of entanglement, maximally entangled pure states are not shareable. The Qubit theory suggests that qualia are encoded in maximally entangled pure states, and for this reason they are private and unshareable. And finally, the fourth question: why does a quali have a particular meaning? The answer is that a pack of information within a cognitive system gradually obtains a particular meaning as it undergoes a progressive process of interpretation performed by a generative model installed in the system. Meaning of information is assigned by a process of interpretation or inference. A system that cannot generate meaningful information cannot generate consciousness. According to the Qubit theory, it is interpretation or inference that turns meaningless information to meaningful information, or turns meaningless bits of information into meaningful bits. Therefore, a system that cannot make inference cannot generate consciousness. There are more to Qubit theory than just these four questions. In the next slides, I will briefly review the most important points of this theory. Some of these points have been explained in more detail in the published paper, and some of the points appear in an upcoming paper. A sensory system performs multiple sequential nonlinear transformations on its sensory inputs, and as a consequence generate a hierarchy of representations at different levels of abstraction. In a sensory hierarchy, a quali or a conscious percept is a maximally compressed representation with the highest quality, while the representations generated at lower hierarchical levels are less compressed and have lower quality. The representation generated at the top of the hierarchy is not only maximally compressed, but also maximally efficient and maximally accurate in predicting the environment. A conscious percept is a representation with minimum variational free energy, minimum thermodynamic potential energy, minimum uncertainty, and minimum thermodynamic entropy. A quali is a maximally compressed, completely random, and non-computable string of meaningful qubits. A sensory system can generate conscious experience only when the thermodynamic depth of its representations goes beyond a certain level. The Qubit theory is in its first stage of development. Apparently, it is not a complete and comprehensive theory. If you read the full article published by Integrative Psychological and Behavioral Science and let me know your comments and feedbacks, I would be very thankful. My email address is majid.beshkar@gmail.com. Thank you very much for your attention.
Hi, my name is Dr. Glen Rein, and I'm going to be talking to you today about phase conjugation in the brain. I am a independent scientist. I left academia at least 10 years ago and pursued my own research with a grant from the Fetzer Institute. And it has culminated in this research about phase conjugation in honor of Edgar Mitchell, who kind of turned me on to the whole topic in the first place. and I would like to honor him in this lecture. If anyone needs to contact me, there's my email address. Please feel free. So, first thing we need to talk about is nonlinear optics, because there are some really unusual phenomena going on in biological systems, and one of which is nonlinear optics, which is described adequately in the physics literature, but these are the various properties. This phenomena is self-focusing. Sometimes it modulates the phase and amplifies the parametric variables, and it is also called second harmonic generation, and it is ultimately called phase conjugation, which is the aspect of it that we're particularly interested in today. So the various methods that are available in the literature for generating a phase conjugation phenomena are based mostly on nonlinear light scattering phenomena, whether it be elastic or inelastic. When it's elastic, the name is given as four-wave mixing, which is another way to generate phase conjugation. And if it's inelastic scattering, it occurs in materials like semiconductors and liquid crystals. And in addition, we have photoreactive materials, which are mostly crystals and gratings, which are physical entities which can be used a bit like in holographic gratings to generate an image of the input. So you still have an input and an output. And that input and output can couple to electrical processes, as in the brain. And of course, phase conjugation mirrors is the way to go. And here's an example of a phase conjugation mirror. On the left, you see an ordinary mirror where the incident light comes in at a certain angle and reflects at a given angle. But in the case of phase conjugation mirrors, the reflected beam takes the path of the incident beam and returns exactly to the origin of the signal. This is a rather unusual phenomena, but it's well described in the physics literature. It was actually first described in 1966, where it was described as Brillouin or Raman scattering, and it was used at the time to transmit images along optical fibers. And as a result of doing that, they discovered that the original image, or the original input light beam, which became distorted when it went through a nonlinear medium, was restored spatially and in terms of phase information. So that was one of its outstanding properties, why it was developed, and why they now know the nature of the reflected wavefront. The beam that bounces back is technically called the complex conjugation of the amplitude of the input light wave. That's a mouthful, but nonetheless, that's a mathematical description. Nonetheless, it propagates back toward the source, as I said, and there is actually a coupling, a rather nonlinear coupling, between the incident wave and the reflected wave. And the two most important properties of this reflected wavefront is that it is time reversed and it is virtual in nature. Two interesting properties, I would say, for the brain. So we have phase conjugation in the scientific literature in certain chemicals, certain molecules, biomolecules even, that have been demonstrated, at least in vitro, to exhibit this unusual behavior. Photosynthetic pigments like chlorophyll is one example. Then we have organic dyes, and the main class of materials are heme proteins, because they contain the porphyrin molecule. And whether now we're talking about common, and biochemists in the audience will know these molecules, hemoglobin, myoglobin, cytochrome C oxidase. So they therefore figured that the function of phase conjugation had something to do with oxygen storage and oxygen transport. In light of the hemoglobin story, it has to do with electron transfer. That applies to all biochemical processes. And in essence, in reference to the photosynthesis, it converts light into electricity. Those are the common ones. I did see one theory proposed that these molecules act as supramolecular antenna. And I will talk a little bit more about the supramolecular nature when we talk about networks. So it turns out that relatively recently, there was an article published in the Journal of Biomedical Optics, which actually measured optical phase conjugation in an intact tissue, biological tissue, rather than an individual molecule. And what they did was they were able to demonstrate that the phase conjugation wave front retraced its steps through the sample back to the origin. And when it did that, it was able to reconstruct the incident light beam. So that's what they actually measured. They measured the degree to which the original source was reconstructed after it got distorted going through the nonlinear medium. And they did that in physical materials, that's classic standards ways of doing it. And they decided, well, I wonder if we could do this in a biological material. They obviously weren't biologists, so they looked around for a biological material, and they probably found a piece of chicken left over from their dinner, and they put that in the machine, and lo and behold, they observed, quotes, the highest scattering as a result of phase conjugation that has been shown in a biological tissue to date. So I got very excited when I read this article. My God, phase conjugation really occurs in the body. In fact, there are several theories that involve phase conjugation way back in the 90s, and then again at the turn of the century by Edgar Mitchell, who theorized the role of phase conjugation in various brain functions, mostly perception. And the idea was that the light from the object, and Peter Marcer was the first person to actually publish this anyway, and he proposed that the light coming from an object that we look at in our perception, in our vision, is reflected off of some kind of phase conjugation in the mirror. He never really specified where the phase conjugation was or what it might be, but we did have a preliminary look at that. The reflected virtual light energy then bounces back, retraces its steps to the source, which is the object we're looking at, and then gets superimposed upon the object. The brain then looks at the combination of the physical object and the virtual object and kind of makes sense out of it. That was the original concept that Peter Marcer proposed. Now, Edgar Mitchell then extended that idea a little bit by talking about virtual holograms that would be a result of the interference patterns that were created by this process. And again, he had a phase conjugation mirror somewhere in the brain, and basically the same idea that Peter proposed, but added the component of a virtual hologram. And he further went on to propose that it would be involved with other mental activities like memory, which of course has been studied in the context of holograms. So, as I mentioned, both of these scientists discussed the idea of having a phase conjugation mirror in the brain, but they had no idea where it was. They did, however, propose in a global sense that they might be involved with neuronal circuits or even spatial temporal patterns. And I'm sorry, but I don't see how a spatial temporal pattern could generate phase conjugation. I'm kind of more in the localized phase conjugation mirror concept outlined here, We've gone over most of these molecules before that are capable of doing it. So clearly, localized phase conjugation mirrors could exist in the brain. In fact, phase conjugation mirrors exist in lots of different chemicals. Well, not exactly phase conjugation, but at least nonlinear optical properties. All these 5 chemicals listed in the far left here, I can barely pronounce, exhibit nonlinear optical properties. So that's a good start, not quite phase conjugation. And then Jibu published many years ago an article about microtubules and also reported the nonlinear optical properties in the sense that they generate second harmonic generation, is what it's called, and quantum coherence. Again, nonlinear properties, but not phase conjugation. The only 2 examples of biological molecules I could find in the scientific literature were bacteriorhodopsin and porphyrins, as I mentioned in the introduction, which actually do exhibit phase conjugation. So since we're not really interested in bacteria, at least in this lecture, we're going to focus on porphyrins. And there are these complexes of 4 different porphyrin molecules forming what's called a metalloporphyrin complex because they're all surrounded by a biological ion, usually magnesium or zinc or copper, as a central coordinating metallic ion. This system is also characterized by pi stacking, which is quite dominant, large channels, and is considered a highly coordinated system, all by itself, the porphyrin complex. Here's an actual picture of it with iron in the center. It's called tetraphenylporphyrin complex, or TPP, in the scientific literature. And it is characterized by highly conjugated double bonds, many delocalized electrons, pi electrons. It's characterized by electron transfer processes, and even quantum tunneling has been observed. So this is a quantum phenomena that we're talking about. Okay, so here's the actual formal paper that was first published in 1984. That was a cosmic year, where they actually measured phase conjugation in these TPP porphyrin complexes. They used rhodamine dye as a laser for the input, and they were actually able to measure what they call the counter-propagating wavefront using vacuum photodiodes. They measured the distortion, then they were able to correct the distortion, and interestingly enough, they also observed oscillatory behavior. This is characterized by certain frequencies, although they were not reported on or calculated. They also reported a relatively high reflectivity, which is a measure of the degree of phase conjugation. In fact, in this particular experiment, it was a 300% enhancement. And this was quite similar to the previous report that we talked about before. So here's the actual initial publication. Since then, there have been several other publications, and they've been able to conclude that there are 3D supramolecular crystalline-like structures, these porphyrin complexes, which then can develop into porphyrin networks. They actually self-assemble, and in the network function, they function collectively. As we said before, they exhibit pi stacking and lots of channels, and not canides, but cavities, which are interesting in their own right because they can trap certain kinds of energy fields and give them exotic behavior. In these particular networks, you have to remember that they're connected primarily by chemical bonds, so they're covalently connected, although there was a report that they also contain hydrogen bonds. And the fact is that they're highly porous. They have these cavities and channels with unknown functions, and they come in pairs, network pairs. Here's a picture of one of those pairs, showing an example of the channel at the top and the chemically linked covalent ions, metal ions, which would be in the center of each of these two complexes. So here's a close-up of what might actually be going on in the brain, where we would have local porphyrin networks, because there are obviously porphyrin molecules in every cell. So if we take some enriched neurons, enriched with porphyrins, we could actually have some kind of an energetic communication between these individual porphyrin molecules. And that's what I'd be talking about today, that these could generate 3D spatial information patterns, which would be non-local in nature. And these could be very much proportional to their specific geometric configuration. I mean, there's almost an infinite number of twists and turns you could use to twist the actual geometric shape. And what I'm proposing here is that this geometric shape is in fact made up of virtual light energy that's generated from the phase conjugation process. That these networks, which are described in the literature, I'm just giving them these energetic properties, assigning these energetic properties to them. They could exhibit quantum information processing, they could exhibit holographic information storage, and they could be involved with some kind of a global information language, you might say the language of geometry, over the entire brain. So that's the background information. So now let's elaborate a little bit more about the model that I'm presenting in this particular lecture. In 2017, I first proposed that phase conjugation occurred in the brain. Well, actually, that had been proposed before, but that the virtual energy part was the critical part, and that in fact that is what the mind is made of. We don't really know what the mind is and let alone what it's made of. So, seeing as the mind has all these exotic properties, it seems logical to me that the mind would be composed of virtual energy. So, what we like to do here is extend the hypothesis of that original model and talk about the networks in a little more detail, and how that the networks can carry virtual energy, and that there are communication channels which allow the transfer of virtual information within a particular phase conjugation porphyrin network and between networks. And furthermore, that these networks can store virtual information in the cavities that we described earlier. Now, we all know probably in this audience what happens when you trap light, laser light, in certain geometries. Well, here we're talking about trapped virtual light. And I have not found any scientific literature exactly what would be the property of a trapped virtual light source, but we can only imagine it would acquire new and exotic and interesting properties. Okay, so now we all know that mind has these capacities of it's capable of mental activity, emotionals, memories, and of course, with this group, the interest is on qualia. And it's interesting that these mental functions have some of the same storage and communication capabilities that I just described and attributed to the phase conjugation mirrors and networks in the brain. So what we're proposing here is that these mental activities and experiences, in fact, can best be characterized as virtual energy in their makeup. This is a rather new concept. Furthermore, individual qualia can be stored in these networks as virtual energy in the way that we described information storage before. Now I'm just attributing the information as the source of the information is coming from these mental experiences and activities. So since these networks can take on unique geometries, and there's probably an infinite number of these geometries, it seems reasonable to propose that the mental activities and experiences can actually be stored in the conformation, in the physical conformation of a network. So although qualia may be etheric in nature, it is stored in a physical structure. This has kind of been a dilemma for many years, although microtubules have been proposed, but there's no evidence that microtubules exhibit phase conjugation. So anyway, the point is that each of these geometric configurations of the networks will store a different mental activity based on the resonance properties, if you will, of the energy, the virtual energy of a particular mental activity, and the geometry and the characteristics of the particular structure that we're talking about. So if there's resonance between the energy and the information, then there will be storage in the geometric networks. Now, there's one other interesting property that I mentioned briefly before for this phase conjugation phenomena is electro-optical coupling. Well, this is particularly important because we're talking about the brain here and all the electrical activity that goes on in the brain. So if we can allow for the virtual networks to couple to the nervous system, then we have a way of communicating and accessing this information, this mental information, which, remember, Freud talked about it as being subconscious and really not accessible to the conscious mind. Here we're proposing that it is rather accessible via this mechanism of electrocoupling. So there it is. There's my model, folks. I appreciate everyone listening. It's been a pleasure talking to you, or myself here, actually, at this conference. And if there's any questions, I'm not available. I'm turning off. And please contact me at this email if you have any. There we go. If you have any questions. Thanks again. Appreciate your listening to my talk, and hope you enjoyed it.
Hi, I'm Greg Scholes from Princeton University. I'm going to talk about quantum effects in biology, and specifically, I'll give some background on coherence effects, and I'll propose some questions for this field. So it's well known that order pervades biology and enables function. You need to look no further than just the synchronous firing of cells in the heart and the function that provides to see an example of this. The question is, on what length scales does this synchronization work? Does it work even at the quantum length scale? Researchers have asked a similar kind of question relating to photosynthesis. They've asked this decades ago, and again more recently. The question comes down to, given that there's hundreds of light-absorbing molecules, for instance, the chlorophylls that you see here laid out in this map, do they function independently or together, perhaps using coherence? So to put this in perspective, any of these hundreds of chlorophylls could absorb sunlight. And if that energy is transferred from chlorophyll to chlorophyll, like somebody passing a ball to somebody else's hands, to somebody else's hands, and so on. This would be incoherent energy transfer, and this would be random. And in a system like this, it would take about 1,000 or 2,000 jumps, random jumps, until the excitation reached the reaction center. Given the high efficiency of the function of this kind of system, researchers have asked, well, is that the case? Is there a smoother path that the excitation can take? And this might involve coherent superpositions, excitation being on more than one chlorophyll synchronously and quantum mechanically. And it's only recently in the last decades that researchers could answer this question or address it experimentally. The key here is that we need to resolve information not just at the probability level, the kinetics level, but we need to resolve amplitude level information in these experiments. To do that, given that we are probing typically 10 to the 20 molecules in these experiments, when we average, we need the ensemble to be choreographed, to be synchronized by some kind of start pulse that starts the experiment. And this is precisely what we can do in spectroscopy if we turn away from the traditional way that spectroscopy works, where we think of it mapping out energy levels or eigenstates of the system. As you see in the absorption spectrum here, different vibronic levels. And instead of that, we turn to a time domain picture where a short laser pulse can excite all these levels. It can't resolve them. What happens? And so here's a picture of what happens. On the left, you see an undergraduate picture of the different quantum levels of a particle in a box. Normally in frequency domain spectroscopy, would map out the energy differences between the rungs of this ladder. On the other hand, if we excite all of these rungs together, and that would involve excitation with a short laser pulse, we produce something called a wave packet. It's a more classical looking particle, if we can call it that, in the excited electronic state that would oscillate backwards and forwards with a frequency determined by these energy spacings. And for vibrational levels of molecules, when we excite the vibrational ladder of a molecule in solution, you can see the result on the right-hand side as a function of time, on the y-axis going down, and the absorption spectrum, the frequency is mapped out on the x-axis by the different colors contained in the probe pulse. So we pump the system with a short laser pulse, produce this wave packet, and we watch it going backwards and forwards from the left to the right side of the potential. And that's what gives these oscillations in the amplitude of the signal with the frequency, in this case, of the vibrational spacings in the ladder. Now, you can think about that same effect in a different way as superpositions of states, and this is where it becomes a bit more interesting. So in the diagram on the bottom left here, we see that our short laser pulse, because it contains many frequencies, it can't discriminate whether it excites level A or level B, two different electronic states. I'll give an example of that on the next slide. Because we can't tell the difference between what we excited, quantum mechanically what we've produced is a superposition state. Famous Schrödinger cat state is often a good way to think about this. And if we can correlate at the ensemble level what's going on, we can actually observe the interference between these possibilities, and that's indicated in these equations by that cosine factor. In other words, there'll be an oscillation in the time domain of this signal with the frequency of the energy difference between levels A and B. These are called quantum beats. So it's an interference effect, but it reports back that we have a superposition. If these levels were not in a quantum superposition, if they were just different uncoupled molecules in solution, we would not see these quantum beats. And that's simply because the clocks are not synchronized. Is the way that I think about it. So here's an example. Here's our two levels. They're labeled HX and LX, heavy hole exciton, light hole exciton. And these are in semiconductor nanocrystal nanoplatelets. You can see them in the electron micrograph at the bottom of the image. These platelets come in variations in size, but their thickness is very uniform. So we have a narrow frequency spread here. And in the pink color, you see the spectrum of the laser pulse. You see it's broad in frequency because it's very short. It excites a superposition of these two states, just like I showed you in the example before, which I'll flip back to. That short laser pulse excites both A and B. It can't pick them out separately, you see, because the spectrum is so broad. And then this is a particular experiment called 2D electronic spectroscopy, which is a bit more sophisticated than the pump probe I showed you earlier, but is a closely related experiment. It allows us a bit more incisively to pull apart this equation 5 that you see here, at least in a simple approach here. We can look at the signals on the diagonal of the plot versus off diagonal and get a different viewpoint into what's going on. Specifically in the off diagonal, you get good resolution of this interference effect, and you see that plotted on the right hand plot, and you see these oscillations here. Those oscillations are happening at the frequency of the frequency difference between the heavy hole and light hole exciton. Now here's the important point of these experiments. You see that we absolutely do see this electronic coherence, this superposition of the two states produced by the short laser pulse, but we lose that synchronization very quickly. 13 femtoseconds is the dephasing time. And that's mostly because there's a distribution of frequencies of electronic absorbers. There's not two discrete levels like you see in this picture here, but a distribution. And therefore, our ensemble experiments lose the ability to pick out that the superposition. Now, turning back to photosynthetic systems, here's a similar experiment on a photosynthetic protein. A very complex system. And what astounded researchers a decade ago was that apparently these superpositions of the same kind that I showed you before, these two levels, the A and the B here, would be of a red and a green molecule embedded in a protein, separated by 20 angstrom. These superpositions seem to last for hundreds of femtoseconds. As you see here in these data, you see those oscillations in real time over a few hundred femtoseconds. It turns out, after a lot of work by a lot of researchers, that this long-lived coherence is caused by a combination of the vibrations I showed you in the first example. It enables, to some extent, this electronic delocalization throughout the protein. It's a much more complicated system than we imagined. And so the long-lived aspect of this, which was the really interesting part initially, is enabled by vibrations which can oscillate for picoseconds, even in these complex systems. So where does that leave us? It does seem that what we might call interesting coherences that would have function on time and length scales relevant to biology are going to disappear quickly because of the complexity of these systems. On the other hand, we do know that synchronization is exploited profoundly in biology. It's not a big leap to wonder if quantum synchronization might be hidden there somehow. We do need to ask what it would do. It may provide new ways to control, to regulate, or amplify processes. And we do need to know where to look for it. Perhaps not in these photosynthetic proteins, but in different places. This is unclear. We'd also need to work out how dephasing can be defeated. The timescale through which these coherences last is limiting. And this is really the problem of entropy. Maybe it can't be beaten. Maybe it can. It's a big question. And finally, can the superpositions be produced in vivo, interesting quantum superpositions, or can they only be produced by technology like short laser pulses? We're encouraged by recent experiments in my lab where it does appear that through separations at timescales, one can produce quantum superpositions or wave packets without short laser pulses. So there's interesting possibilities here. So I'm going to leave you with those questions and acknowledgement here to the funding and my students and postdocs who've been working on this.
Very warm greetings from the Dayalbag Educational Institute. I am thankful to Professor Hameroff and Mr. Aarat Kalra for inviting me to present in this concurrent session on quantum biology at this conference, The Science of Consciousness, TSC 2020. Consciousness studies is an area of research where the Dayalbag Educational Institute excels. But our approach and philosophy is towards the science of consciousness. This evolutionary approach is progressive, adaptive, resilient, giving us opportunity to remain in kinetic equilibrium while pursuing our goals. So today, I am going to present on the possibility of probing the non-classicality of biological systems of nanoparticle protein corona using classical electrodynamic experiments in the gigahertz range. The biggest dilemma that experimental physicists exploring the core of biological phenomenon face today is witnessing the non-classical properties of a biological systems which need not necessarily obey the quantum theory. Because the biological systems are continuously subjected to ambient thermal noise, which makes them to toggle in and out of equilibrium conditions. And these conditions pose a serious question on sustaining of coherence, because coherence seems to be the basic measure of quantumness of any system which is being analyzed for its quantum properties. The warm, wet, and disordered environment in the biological system is far from the ordered conditions which exist in strongly coupled solid-state system where excitations can be completely delocalized over the entire crystal. So the question that I am trying to address today, can purely classical probing methods give insight into the origin and dynamics of biological phenomenon at molecular level? In this study, we investigate through classical resonant approach the interaction of protein, in our case insulin, with zinc oxide spherical particles in the nanometer range and tetrapodal structures in the micron range. And also we have investigated the thermal perturbation and thermal perturbation on the protein functionality or specificity and the effect of natural denaturing agent on the nanoparticle protein corona. If you look at protein dynamics, spatiotemporally, protein can range from few angstrom to few hundreds of microns on the length scale and from few femtoseconds to seconds on the time scale. The dielectric properties of protein varies with temperature, indicating atomic and molecular interactions initiated due to temperature-dependent protein unfolding. The dipole fluctuation depend both on local fluctuation at atomic scale and collective fluctuation at the secondary or tertiary state. So therefore, probing the dipole fluctuation by the measurement of dielectric constant can offer deeper understanding on the interaction mechanism of nanoparticles and proteins and also their unfolding. In the present study, measurement of the spatiotemporal dynamics of slow collective motion of protein is focused because it is the slow motion that is very interesting, where biological activities and the protein unfolding takes place. Nanoparticles, when come into contact with biological fluids, because of their high free surface energy, the proteins are adsorbed on the surface. And this forms the so-called nanoparticle protein corona. To date, most efforts have been devoted in understanding the composition of protein coronas as well as their biological implication. If we look at the literature, then we see that most of these studies emphasize on the change in the synthetic identity of nanoparticle when nanoparticle protein corona is formed. But what about the target-specific functionality of proteins, which becomes extremely important in drug design when nanoparticles are either used as carriers or as preservatives? Because during the adsorption of proteins, their secondary structures can get reconfigured due to strong Coulombian interaction. and measurement of these charge redistribution can be useful in understanding pathological mutations of proteins. In this study, we have used resonant technique to study the interaction of nanoparticle protein corona, and we have used an open-ended coaxial probe. These probes generally are of two types, short monopole type and a long monopole type. So we have used a long monopole probe which demonstrates resonance, and measuring resonance and reflection losses becomes extremely easy through an experimental method, and these resonance and reflection losses are function of medium's material property called permittivity. So in this work, we have a fork type coaxial probe, which is shown here, and it has got a central conductor with two radiating element on the side at 55 degrees. It was seen that at 55 degrees the antenna had maximum directivity, and this was found out by simulation on CST Microwave Studio. So here you see the results of the simulations, electric field, directivity, gain, and Smith chart. So if we look at, so this is the response of the antenna, and here you see all the parameters, design parameters of the antenna that is being designed. In this figure, the resonance, variation in resonance frequency and loss with temperature is presented for insulin, the protein, the denaturing agent papain, and combination of protein and the enzyme, and the interaction of protein with nanoparticles. So in this we see that generally there is an increasing trend of the resonance frequency as we increase the temperature. However, just when these proteins interact with denaturing agent, they tend to saturate at certain frequency. In all these measurements, we see an interesting feature that between 40 and 50 degrees centigrade, there is an inflection point. Either the trend is going from decreasing to increasing or increasing to decreasing. From these resonance frequency and loss measurement, we have calculated the dielectric constant, which becomes extremely interesting for us. So here what you see is the dielectric constant for pure insulin. And here we have when the insulin is interacting with tetrapodal structures, and the last one, this blue one, is for spherical structures. So we see that when tetrapodal structures interact with insulin, they kind of try to conserve the electrical distribution of the insulin, the native or original electrical distribution, dipolar distribution of insulin. However, the zinc spherical structure, they try to disrupt it. So now we see here we have the highest concentration of papain, lower than that, and the lowest concentration. For all the concentration, what we see here is that the papain is kind of impervious to thermal perturbation in the environment, and it also does not change the properties of zinc tetrapodal or zinc sulfide structures, zinc spherical structures. So here the insulin is made to interact with tetrapodal structures, and also insulin is made to interact with papain. And these two graphs are most interesting ones because they show that when tetrapodal structures are introduced in presence of a denaturing agent, how the insulin properties are conserved. So we see here that the tetrapodal structures with highest concentration are able to conserve the dielectric properties. So if you can see here the pure insulin is ranging from 400 to 50, and if we see here for tetrapodal structures at highest concentration of papain, it is also in the same range. So we can easily say that the shape and morphology of the nanoparticle which are being used as carrier or as preservative matter a lot in the formation of nanoparticle protein corona and functional specificity of the proteins. From here, we wanted to understand the molecular dynamics. So we did a small theoretical modeling where we assumed that every protein nanoparticle complex is undergoing or is being acted upon by three forces: binding force, damping force, and driving force. And we solved the differential equation, and from that we got a quadratic equation for the mechanical vibrational frequencies. So theoretical values of mechanical vibrational frequency at different dielectric constant value was extracted, which is plotted here. So we see here that for pure insulin, as we increase the temperature, the mechanical vibration frequencies went down. So if I assume that an insulin molecule is an harmonic oscillator, and if its mechanical frequency is going down, it could be because of the increase in mass or increase in length. So this clearly increase in mass or increase in length only indicates two things, that the insulin is trying to interact with its buffer medium, and the unfolding is taking place as the temperature is increasing. So here we again see the effect on mechanical frequency of papain, and we see that again papain is also, mechanical properties of papain is impervious to the thermal fluctuation in the ambient environment. If we introduce papain to insulin, we initially see that the mechanical frequency goes down, which shows that there is an interaction and larger masses are being formed. However, on increasing the temperature, we can again see that there is a change between 45 to 50 degrees. So at 50 degrees, there is sudden increase in the mechanical frequency. Increase in mechanical frequency means lower mass. So we can say that the papain, which is a natural denaturing, strong denaturing agent, has fragmented the protein into smaller pieces, therefore increase in the mechanical frequency. But on adding tetrapodal structure, we see that this trend of increase in mechanical frequency is reversed, and we see that mechanical frequency goes down. So it only indicates that tetrapodal structure at least prevent the fragmentation of proteins, and there is an unfolding mechanism that is taking place. And same is also measured or is being observed with spherical particles. However, we see that rate of decrease in mechanical frequency with spherical particle is slower than the tetrapodal structures. So in order to experimentally verify our theoretical predictions, an experimental setup was designed. So this experimental setup consists of a spectrum analyzer from Keysight up to 140 gigahertz with a signal generator up to 140 gigahertz. Sample holder was designed using 3D printing, and the metal coating was used using double-sided copper tape. So here we show the experimental and theoretical values. So the black one is the theoretical value, red one is the experimental value. Here by mistake I have written mechanical frequency. It is experimental mechanical frequency. So we see that there is close match. There is a close match. These experimental runs were repeated twice in order to avoid any kind of artifacts. From this, we conclude that morphology and shape can affect the interaction mechanism of nanoparticle and proteins. From this study, it is clear that zinc oxide tetrapodal structures preserve the spatial distribution of charge on the insulin surface. Effect of papain in fragmenting protein is visible through increase in the mechanical frequency. So these are the different runs that were carried out. So here, the change in experimentally observed changes are shown. So then from these mechanical frequency, dipole moment and polarization current density was calculated. In order to confirm our results, zeta measurements were carried out, and through dynamic light scattering, the particle size or the radius of the cluster was calculated. Again, the results are repeated. We see here that when we add tetrapodal structures to insulin papain mixture, the frequencies, the size of the system, the size of the cluster comes very close to the size of pure insulin. So it is again an indication. That using tetrapodal zinc structures can actually preserve the electrical properties of a protein, and therefore, because electrical properties are the measure of polarity of the system, therefore the functional specificity of the protein can be preserved, and the drug can be more effective in target specific actions. So the conclusion that we draw: there is a deep understanding to be gained in tackling the emergence of the essentially classical world of biology from its quantized molecular origin. Classical experimental techniques can give insight into the non-classical phenomenon like nanoparticle-protein interactions and mechanical vibrational frequencies and dipole moment. From the present study, it is concluded that shape and surface morphology can affect the conformation of proteins. Tetrapodal zinc oxide structures were able to preserve the polarity and spatial distribution of the surface charge of insulin and therefore can be effective nanocarriers for insulin. Importantly, the dielectric signals in protein conformation evolves with the temperature of the thermal treatment and is therefore sensitive to the perturbation of local atomic and molecular interaction. So therefore, this technique is effective in characterizing proteins in terms of conformation, stability to temperature, and dynamics with multiple measures such as frequency and temperature variation. So here what you see is the tetrapodal structures which were designed by our collaborators from Technical University in Kiel. So I am thankful to my group members, they are group members, and my collaborators at Germany. I would like to tell here that this work is an outcome of the doctoral research work of Ms. Urvashi, who is my doctoral student. Thank you very much.
Hello, I'm Christoph Simon from the University of Calgary, and I would like to talk about whether quantum entanglement might be able to help explain consciousness. So entanglement is a unique quantum physical property that allows two or more particles to act in a correlated way even when they are separated in space. Bell's theorem states that these quantum correlations cannot be explained in classical terms in any way. And an entangled quantum system should be thought of as one coherent whole, even when it contains many particles. So why entanglement in the brain? What would it accomplish? There's, I think, two main potential answers to this question. On the one hand, there is quantum information processing. So this is a very quickly emerging technology right now that has various advantages over classical information processing. And it's conceivable that nature might also have found a way to harness this quantum advantage, for example, in making computations more efficient or communications more efficient. Then the other potential reason for considering a role for quantum entanglement in the brain is the binding problem of consciousness. So this is a much more philosophical point. That question is basically, how does our conscious experience, which has a very unified sort of holistic character, arise from the dynamics of all these neurons and even all these molecules that we have in our heads? That's very hard to understand from a classical physics perspective, but for entangled systems, there is this holistic character. The whole really is more than the sum of its parts in a well-defined physical and mathematical sense. So that provides another potential motivation for considering entanglement in a neuroscience context. Now, suppose that we are looking for quantum effects in the brain. How could that work? Like, how could they be implemented? What ingredients might nature use to implement such quantum functionalities in the brain? And there we can take inspiration from quantum technology. So we know from quantum engineering that photons are ideal systems for sending quantum information over macroscopic distances. In practice, we do it even for hundreds of kilometers through optical fibers, for example. And we also know that spins, electron spins or nuclear spins, can serve as quantum processors and as memories, even in certain condensed matter systems at room temperature. So maybe they could serve a similar role in the brain too. Now, it's interesting that there are, in fact, photons in the brain. People have seen photon emission from many kinds of cells, and in particular from neurons, for example, in this paper that I'm quoting here. It is thought that these photons probably come from reactive oxygen species, maybe in the mitochondria. An interesting question then is whether these photons could be used for something in the brain. And natural use for them might be for communication purposes. In this context, it's interesting that there are single photon detectors, potential single photon detectors in the brain, namely light-sensitive proteins, so-called opsins. My focus here is on the possibility that the photons could be used for quantum communication, but in fact, even classical communication would also, by light, would also be something new and quite interesting, right, for biology. So the question we asked in this context was whether, so how, if these photons are used for communication, if they are supposed to be used for communication in the brain, then we would need some kind of waveguides, to have the photons, or at least it would greatly enhance their ability to serve as communication carriers if there were some kind of waveguides that allow sort of directed communication from one neuron to another, specifically to another neuron in a targeted way. And so the question was whether the axons that we clearly have in our brains, whether they could serve as such waveguides. And we found that that's encouraging, right off the bat, it's encouraging because the refractive index of axons is in fact higher than that of the surrounding liquid. And if the axons are myelinated, so if they are surrounded by this myelin sheath, then the refractive index is higher still. So light guidance seems really possible. But then the question arises, of course, an axon doesn't look like an ideal optical fiber, so could it really serve as a waveguide despite all these imperfections from a waveguide perspective? So then we made a quite detailed model to investigate that question. So for example, here we modeled the Ravier nodes, because the myelin sheath for real axons is from time to time is interrupted in these Ravier nodes. So we tried to make a realistic model for these Ravier nodes. And we found that transmission can be high despite these interruptions. So that was encouraging. And we also looked at other sort of realistic imperfections. For example, the axons, they are not perfectly circular, right? So the cross-section of an axon can have, they're not perfectly circular. That's true. That's one imperfection we looked at here. Also they don't have a constant cross-section. The cross-section can vary along the length of the axon, so we looked at that. Here I think my next slide has the non-circular cross-section. It might look like this. And in all of these cases we found that it can be okay. If it's within sort of a reasonable range, then photons can still be transmitted theoretically with high probability. We also looked at other things like bends, crosstalk between axons, absorption, which is quite low in the brain actually. So this was all quite encouraging. Then the question is whether there is any experimental evidence for this, and in fact there is some. So for example, there's this paper that was already an important motivation for us, where they showed that Müller cells, which are glia cells in the retina, seem to serve as optical light guides. And these Müller cells, so glia cells are also the cells that make the myelin that surrounds the axons. So that's encouraging. That's quite similar. Here one can see in this experiment they really showed that this cell that is here trapped between two optical fibers, it really guides light through from the end of one fiber to the beginning of the other fiber. So it really serves as a light guide. There's also various more indirect evidence, like a recent one for light guidance by axons. So a recent one is this paper here from 2020, where they showed anisotropic light scattering from axons, and they made the point, in fact, that this suggests that the axons serve as light guides. But more direct experiments would nevertheless be very welcome for this question. So this was the photon part of my talk. Now there is also the spin part. I said that whereas the photons are good for communication. For the spins would be very natural systems to sort of consider for processing of this information and local storage of this information. So what about spins in the nervous system? So there's various points here. In particular, the radical pair mechanism has been proposed and studied quite a lot as an explanation for a bird sensing of magnetic fields, so sensing of magnetic fields by birds, which they do for navigation purposes. So what the radical pair mechanism is that a molecule sort of splits into two pieces, and then each piece has an unpaired electron, but the spins of these electrons are entangled to begin with. And then if there is a magnetic field present, and if there is nuclear spins in these molecules, then this original entangled state has a certain form, a singlet state, can be converted, so this entanglement can be changed and into other types of states, so-called triplet states, and this then affects the reaction rates between these molecules, and hypothetically this is how this magnetic field sensing works in the birds. That's why they would be sensitive to these magnetic fields. So that is one important area of quantum biology where spins really play a role. Nuclear spins have also been proposed as promising systems for quantum information processing in the brain by Fisher in this work here. And then some additional points are that there exist paramagnetic molecules in the body that could serve as interfaces between the spins and the photons. For example, the oxygen molecule is an example, but there's also metalloproteins that could serve as spin-dependent absorbers, maybe. And finally, the point that I want to elaborate on going forward is there's multiple evidence for spin effects in anesthesia, which is, of course, quite relevant for the study of consciousness. So let's talk a bit more about that. So spins in anesthesia. And in particular, I'm going to focus on one specific anesthetic, which is xenon. So xenon is a general anesthetic. It's thought to act in a specific site in these NMDA receptors, but in fact, what I'm going to say could also apply to other targets. So it doesn't. We just had this specific example in mind for specificity, I think. And now there are two very interesting experimental results on xenon. First of all, there was this paper in 2014 which showed that xenon seems to act as an electron bridge because they could see a free electron-like spin resonance signal during anesthetic action, not only for xenon actually, but in particular also for xenon. This was anesthesia of Drosophila, so fruit flies. And then the other experiment that really sort of got me interested in this area was an experiment on mice where they showed that xenon isotopes with no nuclear spin are more anesthetically potent than isotopes with non-zero nuclear spin. And that really is kind of a smoking gun for something. Isotope effects can be really a good indication that something interesting is going on with spins in this case. So we looked at that in more detail, and we developed a radical pair model for xenon anesthesia that explains these observations that I mentioned. So both the isotope effect and the electron bridge effect from the previous paper. So in our model, the nuclear spin of xenon would couple to a naturally occurring radical pair. So this radical pair may have been created by, for example, reactive oxygen species, for example, in this NMDA receptor, or like I said, maybe in the proximity of other targets. So there's a radical pair, and then the xenon acts as an electron bridge for this radical pair, so somehow helping the recombination maybe of this pair. But when xenon has a nuclear spin, then the presence of the magnetic field of this nuclear spin interferes with the electron bridge process, because it flips the spin of one of the electrons in the radical pair, and then they can't react in the same way. So that's the concept, and our model can in fact reproduce the experimental results on the anesthetic potency, the isotope effect, the isotope dependency of the anesthetic potency, using quite reasonable parameter values for the relevant parameters like the hyperfine couplings and the relaxation and decoherence rates. And let me here state it again, that our model is quite general, so it could well also apply to other targets, not just the NMDA receptor that inspired us. So this is in this preprint that we just submitted a couple of weeks ago. Here is an example of our findings. So here one can see sort of the potency, so a comparison between our prediction for what's called the singlet yield from this radical pair, so the reaction probability compared to the measured anesthetic potency, and we want this difference to be small. And what one sees here is that in the regions, so this is plotted for the two hyperfine couplings that we have in our model, the couplings from between the xenon nuclear spin and the two electron spins, and within if you are inside these yellow lines, then there's good agreement between the predictions and the experimental results. And so one can sort of see that we get good agreement when one of the hyperfine coupling constants is large and the other one is close to zero, which then is consistent, in fact, with the idea that xenon may serve as an electron bridge, where like one electron has maybe hopped onto the xenon or has gone into close proximity to the xenon, and the other one is still on some nearby aromatic ring or something. So this is all seemingly reasonable and encouraging for our model. So then the question is again, how would you test this idea of a radical pair mechanism playing a role in anesthesia? And I think the most obvious test is to look at the magnetic field dependence, because our model predicts that the anesthetic potency for xenon, for the xenon isotopes that have non-zero nuclear spin, should depend on the magnetic field. And I cut off the x-axis there, but in any case, in a reasonable range of magnetic field, you would see that the anesthetic potency declines as the field increases. Another thing that would be very interesting to study is whether one can see, so this would be for xenon specifically, but one could also definitely study whether there are isotope effects or magnetic field effects for other anesthetics, and that would then show whether this radical pair model maybe can describe, it's more general, maybe not just for xenon. We don't know that at this stage. So in conclusion, I first talked about how photons guided by axons could enable classical as well as quantum communication in the brain. This was a paper we had in 2016. And then a much more recent result is that the entanglement of electron spins in radical pairs may be what is disturbed by anesthetics, at least in the case of xenon, but maybe more broadly. And now, sort of as a speculative, coming back to my initial motivation about, the relationship, the hypothetical relationship between entanglement and consciousness, like these results are consistent with the idea that large-scale entanglement could be fundamental for consciousness. So, for example, maybe it's some radical pair mechanisms that create the entanglement, and then so between electron spins initially, but then it could be converted into photons and could be distributed via the photons over larger scales, and maybe somehow in this way, complex entangled states that would be related to consciousness would be built up. Of course, this is not conclusive, but at least it's consistent with this general idea. So with this, I would like to conclude and just thank my collaborators and group members who have worked with me on this, specifically on these quantum neuroscience topics.